Hi, I'm Horst von Rakum. I'm from Case Western Reserve University, and I'm excited to welcome you all to OpenBME, a seminar series, online seminar series that's a consortium of 7 schools. These schools here. And I want to point out that openbme.org, at the bottom of this page, is your go-to site for the rest of the seminar series. So at that website, you'll be able to find the speakers for the next several weeks and find connections to things like their abstracts, etc. And this week's seminar is hosted by Rice University, so I want to introduce Professor Gang Bao, who's the chair of Rice University, and he'll introduce the speaker.
Thank you so much. I would like to welcome everyone for joining this OpenBME seminar on genome editing. It's my great pleasure to introduce Matt Porteus to you as the seminar speaker today. Matt is a Sutaja Clerk Professor of Definitive and Curative Medicine in the Department of Pediatrics at Stanford. Matt received his undergraduate degree in history and science from Harvard and his MD PhD degrees from Stanford. His postdoc work with David Baltimore, Nobel laureate at MIT and Caltech focused on developing homologous recombination as a strategy to correct disease-causing mutations, including the AT mutation that causes sickle cell disease. Matt was a faculty member at UT Southwestern, our neighbor, for 7 years before returning to Stanford in 2010. Matt is a true pioneer of genome editing. He worked on genome editing long before it became a big field that now involves thousands of labs, many new companies, including CRISPR Therapeutics, of which Matt was a co-founder. I was very fortunate to get to know Matt actually in February 2008. 13 years ago, I've been very much enjoyed working with Matt over the last 13 years. Without further ado, please welcome Matt for his seminar.
Thank you very much, Gang, for that very kind introduction. And let me echo that the last 13 years collaborating with Gang's lab has been absolutely the most productive years of my academic research career. And it's in no small part because of the multiple collaborative projects Gang's lab and my lab has worked on. I'm really excited to be able to present to the OpenBME. I think at heart, I am a biomedical engineer, even though I'm trained as a pediatrician. And what I'm going to try to convey today is how the precision of genome editing allows you to engineer cells in a way that might create new and powerful medicines, including medicines that might cure currently incurable diseases. As Gang mentioned, I've been fortunate enough in the last decade with the explosion of genome editing to become involved in multiple different companies that are taking advantage of genome editing. But today I'll be talking about everything that's in my own has derived from my own academic lab. So, the 64,000 foot view is that with the development of new technologies comes improvements in the health of people all around the world. And what it certainly is not to minimize the importance of, even simple things like sanitation and clean water. And my condolences for those of you who are in Houston, where that has unfortunately come to the fore. And then in the last few days. But then with the development in the 19th century of antisepsis and anesthesia, making surgical cures possible, the development of vaccines, and of course we're all anxiously awaiting for the SARS-CoV-2 vaccine to be distributed so that our lives can come back to normal. The bread and butter of medicine, of course, is small molecules. And in the last 30 years, the development of biologics, including enzymes and antibodies. But I'm an optimist that that in the next 30 years, we're going to see two types of living drugs really come to the fore. The first is microbiome manipulation, which I'm not going to speak about, but I think has incredibly powerful, could have incredibly powerful effects on people's health. But what I am going to talk about today is using engineering to make cell-based therapies so that we can cure patients. And I think one of the real advantages or potential power of these types of medicines is what I first heard Carl June describe as living drugs. And what that means, of course, is that these go into a body and they're not inert substances. Instead, they are bioactive substances with different pharmacokinetics, pharmacodynamics. They live, they divide, they respond. And in that sense, they really provide a whole new class of medicines. And I think to really realize genetically engineered cells as medicines, we have to have high precision genetic engineering to really broadly apply them. I think even this week, for those of you who are following the gene therapy world, and while lots of new data has to come about, I think uncontrolled integrations, there's going to be some limits to how this could be applied. And so what has developed over the last 2 years in terms of the most advanced form of genome editing, and this is just the most advanced form. There are now base editing and non-nuclease-based editing and prime editing and transposon-based editing and recombinase-based editing. But the most advanced form of precision genetic engineering involves designing a nuclease to make a break at a specific site in the genome. And when that break is created by the engineered nuclease, it can be repaired in one of two fundamental ways: by non-homologous end joining, in which the break gets stitched back together. This is actually a very accurate form of repair, but of course, if the break is repaired accurately, it just allows the nuclease to make another break. And eventually, the cell will create small insertions and deletions at the site of the break. And so this is a way of inactivating genetic elements in the genome. But the process that I'm most interested in is the other way that cells repair breaks, which is by homologous recombination. And the recombination machinery is, normally we'll find an undamaged piece of DNA, usually the sister chromatid, and use that through a copy and paste mechanism to paste the information from the undamaged piece of DNA into the damaged piece of DNA. We can harness the recombination pathway by providing, at the same time we make a break, what we call a donor or template DNA. And for reasons we don't fully understand, but is certainly related to the amount of donor DNA that's in the nucleus, the recombination machinery will use this donor DNA for that copy and paste mechanism. And by designing the donor DNA with the sequence changes we want to make, not only can we target in the genome at a precise location the changes we want to make, but we can also create the exact nucleotide changes down to a single nucleotide level, the changes we want to make. So there are multiple different ways one could harness the recombination pathway as a way of creating genetically engineered cells as medicines. And I'm going to actually go through 3 of them in this talk. The first is to simply make a single nucleotide or a small number of nucleotide changes to correct an underlying pathologic variant. One can think about, and what I won't talk about is one can think about knocking a variant, including a wild type copy of a gene back into its own gene, so it is regulated and expressed under the control of its own promoter. A third use is to use what we call a safe harbor approach, in which a location in the genome is identified that if a expression cassette containing a promoter and a gene is knocked into it, it will have very little perturbation on the cellular function, except that the cell will now express that gene. And then the fourth, and I will talk about this method as it applies to a genetic lysosomal storage disease. And then the fourth idea is to take a gene and knock it into another gene. And in this way, one is rewiring the biology of the cell so that when it's supposed to be expressing the target gene, instead you get expression of the gene you knock in. And I'll talk about how we're applying this for a disease called B-Thalassemia. Now, through many years of work, we realized that if we wanted to apply genome editing to primary human cells, that we had to avoid delivering naked DNA molecules like plasmids into these cells, because primary human cells, unlike, say, HEK293 T cells or K562 cells, have actually retained their cytoplasmic pathogen or damage or danger sensors in the cytoplasm. And since DNA is not supposed to be in the cytoplasm, that's actually a potential sign of a viral infection. When you deliver naked plasmid DNA to the cytoplasm of cells, you activate a type 1 interferon response quite robustly. And that really causes your cultures and your cells to really be very sick. Basically, we give them the flu times 100. So we have had to develop a system in which we avoid using naked DNA molecules to perform the gene editing process. And the system that we have developed is an ex vivo system, so one that's done outside the body, in which we purify our cells of interest. In this case, I'm showing an electron micrograph hematopoietic stem and progenitor cells. We put those into cycle because the homologous recombination pathway is most active in S and G2 of the cell cycle. And then we deliver our nuclease, in which case we use the CRISPR-Cas9 system, and we deliver the CRISPR-Cas9 as a ribonucleoprotein complex with a purified Cas9 protein. We use a high fidelity Cas9 protein that was discovered by IDT, complexed to a what I call an S-cubed guide RNA for synthetic, single, and stabilized guide RNA. And this synthetic guide RNA has end modifications to prevent it from being degraded. This complex is then introduced into the cells by electroporation. Now that gets the break made, but we have to deliver the donor template. And that donor template is ideally, for the classic homologous recombination pathway, is a piece of DNA. And for that, we have co-opted what my colleague at Stanford calls nature's nanoparticle, which is recombinant adenovirus serotype 6. And this virus, for reasons that we don't fully understand, well, evolutionarily has figured out how to deliver its nucleic acid content, which is a single-stranded DNA molecule, to the nucleus of cells without being detected. So that's the system we use. So how are we going to apply that? So the first application I'm going to describe is to genetically rescue a missing function. Okay, and so for that, we're going to focus on genetic diseases in which patients are born with mutations in single genes, and those mutations cause a very specific disease. And the three diseases I'm going to focus on in this talk are sickle cell disease, beta-thalassemia, and MPS I. But there's thousands of such diseases affecting hundreds of millions of people around the world, perhaps probably even more if you think about how many of these diseases are probably never diagnosed. And I will point out that while there are hundreds of millions of people with these diseases, it also affects a broader community of people, because one can imagine that the parents and the close family members and the communities around a patient with a devastating genetic disease are also affected. So the first disease I'm going to focus on is sickle cell disease. And this is a genetic disease. It's probably the most common serious genetic disease in the world, in which a single nucleotide change occurs, in which an A is changed to a T in codon 6 of the beta globin gene. What this results in is a single amino acid change from a charged glutamic acid to a, charged glutamic acid to a polar valine. And the beta globin gene makes a beta globin protein, which combines with alpha globin to form a tetramer. And in the red blood cell, each tetramer carries 4 molecules of oxygen, and it's at near crystalline concentrations in the red blood cell. In sickle cell disease, the single polar amino acid results in a short hydrophobic surface that, after the beta globin delivers its oxygen molecule, results in the exposure of this hydrophobic surface and polymerization through van der Waals forces, converting a red blood cell into the sickle shape. And this stiff sickle-shaped red blood cell occludes blood from getting to the brain, the lungs, the kidneys, and the bones, causing oxygen deprivation in those tissues. and leading to a multisystemic disease. In Africa, where close to 300,000 people per year are born, the median lifespan is in the first decade of life. In the United States, the median lifespan for sickle cell disease is in the mid-40s, despite, contemporary US healthcare. So, talked about that. So we have a disease that is only affecting red blood cells and yet is multisystemic because of the malfunctioning red blood cells. Red blood cells, however, only live for a short amount of time. So if one has to think about a lifetime cure, one has to think about modifying the hematopoietic stem cell that gives rise to the red blood cell for the entirety of our lives. Now, we can cure sickle cell disease by using hematopoietic stem cells from an immune-matched donor, but this is a very complicated process, has a large number of toxicities, and has not been broadly used to cure sickle cell disease for these reasons. And so our goal is to take somebody's own stem cells and genetically correct the mutation that causes the disease, and then give them back as part of what we call an autologous stem cell transplant. And so a large team of people, including Joe Camarena, Danny Dever, Rasmus Back, Annalisa Latanzi, and many others, have developed a system in which we use the RNP. Actually, we use a guide RNA that Gang Bao's lab discovered that makes a cut very close to the mutation that causes sickle cell disease. And it also does not cut a nearby, very homologous gene called delta globin. And then we design our donor template will correct the change, but also introduce silent changes in the targeted allele to prevent recutting of the sickle cell, of the beta globin after the correction occurs. And using the system, after a little practice, we now routinely get in the lab between 60 and 75% of the alleles corrected. So that means many of the cells have both alleles corrected. So what we need to show now is that once we've done the correction, do they make red blood cells that make the non-sickling hemoglobin A, and do they retain their hematopoietic stem cell properties? So to check the first about what hemoglobin they make, we take CD34 cells, which is a marker of the stem and progenitor cells in patients, correct them using the process I described, and then in the laboratory, differentiate them into red blood cells. And we can measure then the amount of either hemoglobin S or hemoglobin A. And you can see after we take CD34 cells derived from sickle cell patients and correct them, we convert from making entirely hemoglobin S to essentially all hemoglobin A. And so this is now really very close to what people without any evidence of sickle cell disease would have. The next thing is what do after this manufacturing process, which is relatively complicated because it means we have to purify the cells, we have to culture the cells in cytokines, we electroporate them, we add a virus. Do they still retain their stem cell properties? The only definitive way of proving whether they have retained their stem cell properties is, of course, by putting them back into a patient. But the FDA won't allow us to do that unless we show some evidence that they do retain their stem cell properties. And it turns out that depending on the source of the stem cells and the mouse you inject them in, you get different results. But the key is that when we take mobilized stem cells from sickle cell disease patients, take them through our manufacturing process, and then transplant them into an immunodeficient mouse, so a mouse that does not have an immune system to reject the human cells, we find that average correction frequency is about 30%. And that exceeds the 5 to 20% level that has been proposed as potentially curative for patients. So we believe that we have an efficacy endpoint. What I'm not going to describe, sorry, let me just go back, is we have characterized the guide RNA and the process extensively for frequencies of indels, frequencies of translocations, and have found that while we see a small frequency of indels and an off-target site, again, an off-target site that Gang's lab helped identify, that indel site is in a site that is not close to any gene and of no known functional significance. Now, recently, we took the same process and modified mouse HSCs and put it back into mouse to see if if the fall in gene-corrected cells was the result of this xenogenic model of putting human cells into a mouse. This was done by Adam Wilkinson in Hiro Nakauchi's lab. And so what he first did is we helped him design a guide RNA to cut the ROSA26 locus, the sort of universal safe harbor in mouse cells. And into the ROSA26 locus, he inserted a expression cassette for GFP. And what you can see is in the laboratory, he gets somewhere between 17% GFP positive cells to, here we go, between 17 and 30% GFP positive cells in mouse HSCs. Now, we get much higher frequencies in human HSCs, but this is because we've optimized the process in human cells and have not optimized in mouse cells. Yet, nonetheless, we get reasonable amounts of targeted integration in mouse HSCs. And when Adam transplanted these green mouse HSCs into mice, both in a primary transplant and a secondary transplant, what you can see, I think most notably in the secondary transplant, which is really testing, have you modified a true HSC, is that the percentage of green cells is equivalent to the percentage that went into the mice, around, again, that 20-25% level across the red cells, platelets, myeloid cells, B cells, and T cells. So a multi-lineage targeted integration. So this was very supportive that we could target true HSCs using the system. And then Adam applied that directly to a mouse model of sickle cell disease called the Townes mouse, in which the mouse globin genes have been replaced by the human globin genes, including the sickle globin gene, human sickle globin gene. And again, what you can see is the allele correction frequency in the test tube is around 20%, lower than the 60 to 80% I showed you earlier. But nonetheless, when he transplanted these corrected Townes mouse sickle HSCs back into the Townes mouse, you can see that there was a dramatic increase in hemoglobin A and that there was gene correction across multiple lineages, including myeloid B and T cells. And so this demonstrates, again, that we have modified HSCs and that when we modify a small number of HSCs, we can get robust hemoglobin A expression. So with this data, the safety data that I described, and data showing that we can scale the process up in a GMP facility, which I haven't shown, we actually got IND approval in late last year to proceed with a clinical trial into which we will harvest CD34 cells from first adults. I show a child here because ultimately we want to treat children before the destructive effects of sickle cell disease manifest themselves. We will manufacture them in a specialized GMP facility, as I described, using reagents from the various suppliers, and a shout out to all of them for helping support us in this process. And after the manufacturing process is completed, the cell product will undergo a quality control, including cell counts, cell viability, sterility, and importantly, a measure of what the frequency of correction is. And if it passes our quality control, then the patient will come back to the hospital, receive full dose busulfan chemotherapy to ablate the endogenous stem cells that would make sickling red blood cells, and we will transfuse or transplant our drug product into the patient, and then keep our fingers crossed that this gene-corrected cell product reconstitutes the hematopoietic system of the patient, including making red blood cells that no longer sickle. And we hope to start enrolling in this study in the next actually few weeks. We'll let you all know. Okay, so I just talked about how we can use stem cell engineering to rescue a missing function. I want to describe another application of rescuing a missing function, but using recombination in a different way and applying that to beta thalassemia. And I'll get into what we're going to do here. Now, and I'll describe beta thalassemia in a second, but the general point is we're not going to directly correct the gene mutation, but instead we're going to harness the targeted integration of a wild type gene into another locus. So let me dive right into what I mean by that. So beta thalassemia is a cousin to sickle cell disease. It's a cousin because mutations are both in the beta globin gene itself. But unlike sickle cell disease, where the mutation causes a dysfunctional beta globin protein, in beta thalassemia, the mutations cause a lack of expression of the beta globin protein. What this leads to is that the tetramer, which I described earlier, consists of 2 alpha chains and 2 beta chains, no longer has beta chains. And so you get an anemia from the lack of beta chains. Moreover, the alpha chains form aggregates and precipitate and cause the developing red blood cells to die. So you end up with a severe anemia, and in the most severe forms, patients die in the first year of life because of their anemia. So we have 2 problems. We have too little beta globin and too much alpha globin. So our idea here, the hypothesis is that we could target beta globin into alpha globin. Now, this might seem like it would be impossible, but we take advantage of the fact that there are actually 2 copies of the alpha globin gene, alpha 1 and alpha 2. And so what we know is that if people are missing one alpha gene but not the other, they are still very healthy. So the idea then is to use gene targeting to replace one alpha globin gene with a beta globin gene, and thereby normalize the ratio of alpha to beta, or at least almost normalize the ratio of alpha to beta, as well as creating enough hemoglobin that there's no longer anemia. So what we had to do first was to find a guide RNA that would cut one alpha but not the other. Because if we cut both alpha genes, we would simply convert somebody from beta thalassemia major to alpha thalassemia major. So we wanted to only cut one alpha gene. And I'm going to just have you focus over here on guide 5, which was able to create indels in alpha 1, but not in alpha 2. I will note that these indels, this guide RNA is in the 3' UTR because the coding region of alpha 1 and alpha 2 are absolutely identical. So it would be impossible to design a guide RNA that distinguished between alpha 1 and alpha 2. But this one distinguished because there were 5 mismatches between the 2 genes in the 3' UTR. So then Kyle Cromer, the postdoc in the lab who led this project, measured the frequency of what we call whole gene replacement versus cut site insertion. So inserting a gene right at the site versus replacing the entire gene next to it by making a cut at one end. And so we didn't know if whole gene replacement was going to work or not. But what you can see is if you measure the frequency of whole gene replacement versus cut site integration, you can see that at least at the alpha 1 gene using guide RNAs, whole gene replacement is at least as efficient as cut site integration. So it gave us then the opportunity to replace the GFP cassette with the beta globin gene. And so again, what Kyle did is he made up various numbers of different replacement constructs, including ones that contain different 5' and 3' UTRs and different lengths of the homology arm on the 5' end, and found that the best construct was one in which the 5' homology arm was longer than what we normally use, 800 base pairs instead of 400 base pairs, and he could get approximately 37% integration of the beta globin gene into the alpha 1 gene. So then we wanted to see, well, do we start rescuing the hematologic defects? And we actually had a challenging time obtaining beta thalassemia patient-derived CD34 cells, but we had ready access to sickle cell patient-derived CD34 cells. And so what we could use these cells, because when we differentiate red blood cell into red blood cells, the hemoglobin S molecule migrates differently on HPLC than the hemoglobin A. And what Kyle was able to show is by measuring the ratio of hemoglobin A to hemoglobin S and plotting it against the percent targeting, that there was a direct correlation, and you can see the slope is 0.85 or 0.86 between the percent targeting and the fraction of hemoglobin A that is made compared to hemoglobin S. And you can see again with the long homology arms, we're getting more targeting and more hemoglobin A. Eventually, we were able to get beta-thalassemia patient-derived CD34 cells from John Tisdale and Vivian Sheehan. And when we looked at the mRNA levels of alpha to beta, we can see that after gene targeting, in which we were getting 50% whole gene replacement, the ratio of mRNAs was approximately equal. And when we then measured with Vivian the protein levels, we found that the ratio of beta to alpha was 0.5. For the non-hematologists out there, I will say that those people out there who have beta-thalassemia trait, so one gene that's inactivated for beta globin, but one gene that is functional for beta globin, also have a beta to alpha ratio of 0.5. And there are millions of people in the world with beta-thalassemia trait, many of whom have no idea that they have beta-thalassemia trait. So it's a very healthy condition. Now there is a mild anemia, so they're unlikely to win marathons or endurance races, but for day-to-day life, they're very healthy. So now what we've done is we've, again, just as an example to an engineering audience, is about how to sort of rewire stem cells such that they create progeny that have a different character, but that therapeutic character that is beneficial. So the last two examples I want to focus on are not focused on rescuing a missing function, but again, extending the precision of editing and combining it with the principles of synthetic biology. That is, we're going to engineer cells to do something that they don't normally do. And the first application of this will be for a disease called mucopolysaccharidosis type 1. And in this genome editing strategy, we're going to use the safe harbor approach. And I'll explain to you why we're going to use the safe harbor approach, in which we're going to target a promoter and the gene of interest into a specific location in the genome that we have analyzed to being potentially safe and not cause any genotoxicity. This project was led by Natalia Gomez-Espina in my lab, who now is an assistant professor at Stanford. And it was recently published in Nature Communications. So mucopolysaccharidosis type 1 is a genetic lysosomal storage disease in which patients are missing an enzyme called iduronidase. And there are variant forms of this disease. And the most severe form, in which patients have very low enzyme levels, it's a multisystemic disease that causes CNS impairment and multisystemic disease, because what this enzyme is supposed to do is break down glycosaminoglycans into smaller, more metabolites that can be, again, cleared. If you're missing this enzyme, those glycosaminoglycans build up in the tissues, causing significant issues. What's interesting, though, is that there are patients who only have approximately 1% of enzyme, but have much milder forms of the disease, including having none of the CNS manifestations. So a little bit of enzyme can go a long way. Now, currently, the way this disease is treated is in two ways. One is you can give the enzyme every week as a biologic. Now, this enzyme can process the glycosaminoglycans in the liver and spleen and can deal with that issue, but they do not cross into tissues like the eye or the bone or the brain very easily, and so they don't deal with the really severe manifestations. But it does show that enzyme from outside the cell can get inside the cell and start processing these GAGs. The other approach is actually really surprising, but it works, which is you can do an allogeneic stem cell transplant in which you take the hematopoietic stem cells from somebody who doesn't have the disease, transplant them into somebody who does have the disease. the progeny of the hematopoietic stem cells, most importantly the macrophages, will migrate into the tissues, make a small amount of enzyme that will be secreted into the extracellular space and be taken up through a process called cross-correction by the cells that are missing the enzyme, and we will get a low level of metabolism. And what is really important is that if you use people who have both copies of the gene, MPS I gene intact, they are better at ameliorating the symptoms than cells that only have one copy. So there's a dose response here. So what Natalia hypothesized is that instead of using an allogeneic transplant and normal levels of the enzyme, is we could take the patient's own cells, genetically engineer them through safe harbor gene addition, such that the progeny make more super physiologic amounts of enzyme, such that they, when the cells migrate to the tissues, they can cross-correct multiple tissues. And so she could demonstrate that her and her team could demonstrate they get it between 40 and 60% targeted integration into the CCR5 gene, the gene we've chosen to be a safe harbor because we know that there are healthy people out there who are missing both copies of the gene. And we can show that high levels of enzyme are expressed using a PGK promoter. In fact, 50-fold more enzyme is expressed than normal. So we hope we've supercharged or made these cells more potent. And really excitingly is that when she transplants these human cells that are expressing more enzyme than normal into an immunodeficient mouse that also has MPS I, she can see that the glycosylaminoglycans that are at high levels in the urine start to decrease over time in the mice that are transplanted with these human cells. And importantly, some of the phenotypes, the clinical features in the mice that mimic the human disease also improve, including that the bones now come back to normal thickness. In the disease, the bones are thick because of the buildup of these gags. And really importantly is that she showed that multiple behavioral phenotypes improve. So you can see that the red lines now are more like the heterozygous normal controls than the diseased blue lines. There isn't full correction in all phenotypes, but there is certainly substantial correction in many behavioral phenotypes. And there is a reduction of inflammation in the brain because of, again, the migration into the brain of macrophage derived from the bone marrow that then deliver this enzyme and reduce, although still not completely eliminate, the microgliosis that is found. So Natalia is now further enhancing the potency of these cells and trying to develop even better ways of getting migration of her super potent macrophages into the brain to really fully rescue the defect. And the final application I'm going to describe, and hopefully we'll leave enough time for questions, is again a synthetic biology application. And this was a project that was recently published in Nature Biotechnology. You'll notice one of my key collaborative authors there, Gang Bao. And it was done by Volker Wiedking, talented MD postdoc in the lab, and in collaboration with James Patterson, who really had the initial idea and brought it to us about, hey, could we actually get it to work? And the idea is to use genome editing to engineer the metabolism of cells. And specifically, the idea is to generate human auxotrophy or human cellular auxotrophy. Auxotrophy is a state of the cell in which they require a downstream nutrient to grow. So the principle is if you have a metabolic pathway that you can use genome editing to inactivate one of the enzymes such that this metabolic pathway can't work and thereby the cell will be dysfunctional, but you can rescue that metabolic pathway by providing a downstream nutrient. So it gives you control over the cell behavior. And the pathway that we studied and James identified was a pathway for in the metabolism of uridine, who have mutations in UMPS, and they can be rescued by oral administration of the downstream uridine, by downstream uridine triacetate. And so again, the idea then is, can we engineer cells by knocking out UMPS and then give us the control of those cell behavior by either providing or withholding uridine triacetate. And so Volker showed that both in primary human T cells or in human embryonic stem cells, that if you made knockouts in the UMPS gene and you withheld the uridine, the cells were unable to proliferate. But if you provided uridine in the culture, you could rescue the proliferation of these cells in T cells essentially back to normal, and in ES cells back to normal. Which is great in vitro, but how might one want to apply this as a cellular, as a control of a cellular medicine? And so there were three potential ways we might use this. One would be if you gave a cellular medicine and for unexpected reasons it caused a transformation, one might be able to control the transformation by removing the nutrient from the diet. The other is as people are developing induced pluripotent cell-based cellular regenerative medicines, the concern is, again, about transformation or the development of teratomas. And so one could make these cells dependent on a nutrient that one could regulate. And then finally, as we all know, engineered T cells are transforming how we think about treating cancer, but we're always concerned about those engineered T cells causing problems. So can we control them in a way? And I'll show you that each of these three things we can do. So the first example is to take a leukemia cell line, make it UMPS knocked out, grow it either with or without uridine triacetate in the chow. And you can see without uridine triacetate, the leukemia doesn't develop. With uridine triacetate in the chow, it does develop. And with uridine triacetate, the leukemia develops. And then if you withdraw uridine from the diet, what you see is that the leukemias will melt away. So it gives a proof of concept that one can control a leukemic transformation by metabolically being able to control it, or the gene. In terms of controlling teratomas derived from ES cells, very similar model where teratomas were created in mice from UMPS knockout ES cells. They were allowed to grow to a certain size, and then they were either kept on the uridine triacetate supplemented diet, or the UTA was removed from their diet. And one can see that when the UTA was removed from the diet, the teratomas shrank dramatically, although they did not disappear. And this becomes both an interesting bug and perhaps an interesting feature, because it means your cellular product is still there if one wanted to reactivate it, but it is under control because it is unable to proliferate. And then the final example is in T cells, where again, a very similar model, creating UMPS knockout T cells, injecting them into mice, allowing them to grow, and then removing the uridine triacetate from one cohort and keeping uridine triacetate in the diet of the other cohort. And one can see that in the mice that have the UTA food, the T cells persist, but after a 2-week period without uridine triacetate, the T cells disappear. Again, this is a feature in bug that it is a slowly responding system. What this would mean is that if a patient forgot to take their UTA one day, it would not be a total disaster. But it also means it's not a system that would allow you to rapidly eliminate T cells if they were causing an acute toxicity. So this is a slide just comparing how engineered oxytrophy compares to some of the more classic suicide genes in terms of the pace of the action, the reversibility, the probability of escaping, which is quite easy or is quite measurable with suicide genes, but is impossible because we've made double knockouts with engineered oxytrophy and some of the other features of how the system might work. So the last thing I want to end with is just, I'm speaking to a really great audience of engineers, and trying to maybe inspire you to join me in the effort of how we might be able to take really complicated cellular therapies that might be available at our local institutions and get these therapies to people in all parts of the world where there is a tremendous suffering with these genetic diseases. I'm not proposing that this is going to be an easy step, but I am also suggesting that cellular therapies are such a new type of medicine that we have the opportunity to think about how we can use bioengineering to bring these therapies to multiple parts of the world. With that, I want to thank you for the opportunity to talk to this great audience. Many, many names on this list because of the many, many contributions, both in my lab, my collaborators at Stanford, collaborators in our GMP facility, funding from a broad range of sources. And where are you, gang? You're very much on here. You should be right. Where are you? Anyway, and a long time collaboration with Gang. There's Gang Bao and Karen Lee at Rice. Long time and incredibly productive collaboration with Gang. His creativity, his motivation, and his anything's possible attitude is really inspiring. So with that, I'll stop talking, and I see some questions have been accumulated in the chat and the Q&A, and so we'll go to try and answer those. Thank you.
Thank you so much, Matt, for the wonderful talk. Very exciting work. So also congratulations for the clinical trial.
Thank you.
Keep us informed. Let's go to the Q&A now. The first question was if this type, the type 1 interferon response mediated by the C-gas system. So that's first part of the question. And if so, the small molecule inhibitors of the system improve genome editing efficiency?
Yeah, that's a fantastic, that's a really interesting question, and one that we want to study. As Gang mentioned, I was at UT Southwestern for a number of years, and James Chen, of course, I couldn't help but pay attention to all the great work he was doing around the C-gas sting system. So I think the hypothesis you propose is one that's very interesting. We don't know yet if the C-gas sting system is directly involved, but I think it's likely to be involved. And I think then whether you can find inhibitors as a way of enhancing genome editing is really great. So, if you have the opportunity to do it, I encourage you to do that. We're also starting to look at that as well.
Thank you, Matt. The next question actually is what you mentioned, Matt.
Yeah.
Bluebird Bio suspended their clinical trial a few days ago on sickle cell gene therapy due to a patient treated 5 years ago but developed AML. So the question is, could you comment on this event and whether you see a possible path without sickle cell gene therapy based on genome editing could also lead to AML?
Yes, it's a really great question, and one that myself and many others have been having a lot of conversations about. First of all, I want to, I think we need to applaud Bluebird for being very cautious and suspending their clinical trial. There is still very little information that we know about both the patient with AML and the patient with MDS. What has been publicly disclosed, and I know no more than what's been publicly disclosed, is that in the AML, they can detect an insertion. What we need to know now is where that insertion is and how that may or may not have contributed to the development of AML. Certainly, one of the differences between targeted integration and lentiviral integration is that in lentiviral integration, we don't control where the integration occurs. In fact, we know that lentiviruses have a predisposition or a bias to integrating into the introns or the first intron of transcriptionally active genes. So in fact, it kind of goes exactly where we don't want it to go. On the other hand, it does facilitate expression. The other, so we need to learn more about what's going on. The other issue is that, and I mentioned in our trial as well, is that we, in order to get the desired effect of our genetically engineered cells, whether it's a lentiviral transduced cell or whether it's a genome edited cell, we have to give the patients busulfan. Busulfan is a very old chemotherapy drug that causes DNA damage, and it is known that at some low frequency, busulfan can induce a cancer, including myelodysplastic syndrome and occasionally AML in patients. So we're going to have to learn whether these patients might not have a lentiviral problem, but have a busulfan problem. If they have a busulfan problem, then it applies to any use of busulfan, but it could be more exacerbated in sickle cell disease. And it suggests that the efforts at developing ways of creating bone marrow space without having to use things like busulfan is really going to be important. And of course, there are a number of academic investigators and companies focused on how can we get rid of busulfan from bone marrow transplants. And of course, now we're even more interested in that. So thank you for the great question. So anonymous, is the sickle cell disease therapy curative or must it be periodically repeated? The goal is, of course, for it to be lifelong and curative because we've modified a lifelong hematopoietic stem cell. We know from allogeneic stem cell transplants that it is lifelong because we can transplant a true long-term hematopoietic stem cell. It is only through long-term follow-up of patients, and we have done a lot of testing to see if we will get curative therapy, but the only real test will be to follow patients over the course of years, if not decades, to see if this would have to be repeated. Now, since it's an autologous product, in principle, one could manufacture the product again, but going back to the busulfan question, I would not want to be giving patients busulfan at the doses we give over and over again. So we really need to shoot for one-shot curative therapy.
So Matt, my own question actually is, given your involvement with CRISPR therapeutics, would you compare the two approaches? One is to use HbF, the other is gene correction, right? Of course, we hope both will work, but could you comment on the differences and pros and cons?
Yeah, let me just give a little bit more background. So what Gang is referring to is another strategy to potentially cure sickle cell disease is to upregulate fetal hemoglobin. Fetal hemoglobin is a kind of like beta globin. It is the globin that we express when we're babies. And we transition from expressing fetal hemoglobin, hence its name, to adult hemoglobin, hemoglobin A, within the first year after birth. What people have found is several things. One is that fetal hemoglobin is an anti-sickling hemoglobin. It interferes with the polymerization process I discussed. And studies of patients who have variable levels of fetal hemoglobin have shown that a little fetal hemoglobin, perhaps as little as 20 to 30%, can interfere with the sickling process. And actually there's a medicine, an old school medicine called hydroxyurea that in part acts by upregulating fetal hemoglobin. So the other line is that people have used genetics and molecular biology and animal studies to demonstrate that the master regulator of the switch from fetal hemoglobin to adult hemoglobin is a transcription factor called BCL11A. And again, numerous investigators, primarily led by Dan Bauer and Stu Orkin at Harvard, however, demonstrated that there is an enhancer in the BCL11A that turns the transcription factor on in the red blood cell lineage. So we call that the erythroid enhancer. And what they showed is that if you inactivate the erythroid enhancer, you can reduce BCL11A in red blood cells and upregulate fetal hemoglobin. So the CRISPR therapeutics trial has shown that they can design a CRISPR guide RNA to disrupt the erythroid enhancer in patient cells, and that when they give these cells back to patients with either thalassemia or sickle cell disease, that they see a significant upregulation in fetal hemoglobin to levels that would certainly be predicted to block any of the sickling process. So what we have here is what I would say is an indirect approach, but all it requires is to create an indel. You do not require having to harness the homologous recombination pathway. So on one hand, the indirect pathway is simpler because you only have to create an indel, and indels can be created throughout the cell cycle. On the other hand, I'm of course biased towards the gene correction approach, which is, while it's more complicated, is a direct correction of the disease-causing mutation. So I think it'll be very interesting to compare how patients do, and it'll be very interesting to see whether the simpler manufacturing might make this more widely available, or can we make the manufacturing of gene-corrected cells as simple as the sort of hemoglobin F upregulation manufacturing. Finally, I will also just say that, and I think I showed in this talk, that there are many other applications of homologous recombination one can use. There are very few diseases, although sickle cell disease and beta thalassemia are two of them, that you could treat by making indels alone. So hopefully that gives a fair comparison. Okay.
Given the time, there's another question.
Yeah,
given the high cost of gene therapies, what is the expected accessibility of the treatment of sickle cell disease with gene editing together using, of course, as you point out, using AAV as a way to deliver those, especially as this primarily affects persons of African origin.
It's a you know, and that's the sort of question that I ask myself as well. And I think, and Gang and I have had many discussions about this, and you know, again, I really appreciate his optimism and ambition. I think the first step to improving accessibility is first to create a supply chain of reagents that is more reliable and cheaper. Right now, in part, the cost of therapy is driven by the reagent supply chain. So I think that is something that can be solved. The second thing, again, is a solvable problem, is to take a very complicated manufacturing process that has to be done in specialized facilities that cost millions of dollars to build and millions of dollars to run, and put it into a closed manufacturing system. So simplify the manufacturing so it's automated. And again, I think there are devices out there, such as the Miltenyi Prodigy, that give a sense that such closeness to manufacturing is in the near future. I think there are going to be other things we're going to have to solve, but I think there are, I'm a believer in iterative improvements that get you there, that one does not need to solve the entire at once, but it is important, in fact, it's really important not to get discouraged by the big problem, but instead chip away, and chip away. And eventually, I think we can get there.
Thank you so much. Thank you again, Matt, for the very stimulating, wonderful talk. Would like to also thank, of course, all of you in the audience. I would like to thank host and Steve from Case Western for helping us setting up this seminar. Now I would like to invite students, trainees who received the invitation to have a discussion session with Matt. Thank you so much.
Thank you very much. And thank you to the person who posted the link to a organization that advocates for global accessibility. I really will take a look at that myself. But again, thank you very much, and look forward to interacting in person in the future.
Thank you, Matt.
Thank you.